Hello, my name is Dr. Andrew Matamoros, and welcome to today's online lecture covering spinal cord injury. Today we are going to shift gears away from the CDC, where we're usually discussing diseases, and move on over to the NIH at NINDS, which is the National Institute of Neurological Disorders and Stroke. To start us off, we're going to see some statistics from NSCSC, the National Spinal Cord Injury Statistical Center. Here we can see that's where I'm getting these numbers. So currently there are about 294,000 people living with the spinal cord injury in the United States. And each year we add about 17,800 people to that list. And it's important to note here in the title that there is no cure for spinal cord injury, meaning that these people essentially have to live with and manage the symptoms of a spinal cord injury. So what are some of the risk factors that put you at a higher risk of sustaining a spinal cord injury? Well, first off, it's being a male. So SCIs affect a disproportionate number of men, and in fact, only 20% of females are part of this SCI community. Age seems to play a part in your susceptibility or risk factor of incurring a spinal cord injury, with ages 16 and 30 having the highest incidence. Whereas we can see here some discrepancy in these numbers where the average age of time of injury is 43. So why is that? Well, it's because over the age of 65, falls are a big cause of most of the SCIs in adults. So that's where we see this averaging out at 43. But really, you're at highest risk between the ages of 16 and 30 and over the age of 65. Now, another thing that's going to put you into this risk category is just risky behavior in general. So things like diving into too shallow water, playing sports without protective safety gear, and just overall taking the general precautions that are necessary to protect your spine. We also see that motor vehicle crashes are going to be the leading cause of spinal cord injuries for people who are under the age of 65. And I actually think right here it's this risky behavior that adds a little bit of a socioeconomic factor on your risk for SCI. So, if you can afford to go skiing, diving, taking on sports games where you're having high impact and putting your body at risk, some of these things can be tied to socioeconomic standards. So I think that's where we see some of the race and ethnicity differences with such a high incidence in the non-Hispanic white population. This could be linked to potentially riskier behaviors, most likely due to access of money and being able to partake in those risky behaviors. And then also we can see here what qualifies this for a topic in this class, disease of the human body, is that spinal cord injuries can also be a result of a disorder, such as a bone or joint disorder. So things like arthritis or osteoporosis can actually put you at a greater risk for an SCI. Now we're going to look at how spinal cord injuries are caused. We'll see that motor vehicles top our list. They actually contribute roughly half of the new spinal cord injuries each year. Next, we'll see falls, again with individuals over 65. Acts of violence making up 13%. This includes gunshot and knife wounds. Sports and recreational injuries also make the list, with about 10% of SCIs being accounted for by these injuries. It's interesting to note that alcohol is involved in about 1 in every 4 SCIs. And finally, we see how spinal cord injury has made the list for our class, diseases of the human body, because we see here that diseases can in fact cause an SCI. So we see that cancer, arthritis, osteoporosis, and inflammation can lead to an SCI. And cancer and inflammation we both have gone over. And we should be aware that tumors can occur anywhere, and also that inflammation is very damaging when uncontrolled. Spinal cord injury is life-altering. Just how altering it's going to be will depend on the level and extent of the lesion. Here we see that spinal cord injuries are actually quite diverse. Now, what we can break this down to into general terms are the words paraplegia and tetraplegia. Now, paraplegia and tetraplegia simply refer to where on the body the individual's been affected. So paraplegia demonstrates that from the legs down, the individual's affected, and for tetraplegia, we can see from the neck down. So in this case, both the arms and the legs are being affected in some way with respect to their sensory and motor function. In this image to the bottom, we can see injuries can even be further categorized by the location to which they're incurred on the spinal cord. So for example, a C4 injury takes place in the cervical, the fourth vertebra, and this gives rise to a tetraplegia. This would be compared to a T6 injury that takes place in the thoracic region at the sixth vertebra and results in a paraplegia. So we can see that with this diversity in symptoms, or diversity in injuries, we also get a diversity in symptoms. Now, the first symptom we always think about is the loss of movement. So individuals who end up in a wheelchair and can no longer move their legs. However, we can see here that a much longer list of symptoms are being reported. So all of these things are going to affect spinal cord injury patients in different ways that are, again, dependent on the types of injuries that they occur. But it's worth noting that treating these symptoms can often improve the patient's life, even more so than just simply focusing so much on movement. Now I'm showing some tables here that depict the marital status of spinal cord injury patients after years of incurring their injury. So we can see that the divorce rate 40 years out has increased quite a bit from 21% to 8% compared to when they first received this injury. So the purpose of showing this table is just hit home the idea that not only are the spinal cord injured patients affected physically and emotionally and personally, but these things are also affecting their livelihood, people around them, even we can see their occupational status. So after this type of injury, we can see that 40 years out, only 32% of spinal cord injured patients are employed. See here that we're comparing different degrees or severity of injury. So we have our high tetraplegia as compared to just the paraplegia. And I want to hit home this idea that the more injured a person is, the more they're going to spend at the hospital. And it's not just in this first visit, but also each subsequent year. And we can see over to the right that when the person is going to incur this injury will also impact just how much they spend in healthcare costs. So if someone is injured at the age of 25, they could expect to spend upwards of $5 million through their lifetime, whereas someone injured at 50 years old is going to be spending about $2 million. So spinal cord injury does not always result in a loss of life. These patients are quite, healthy and happy after their injury with relatively speaking to other types of injuries out there, and they can live a long life, and so they can also expect to end up paying a great deal in medical costs because of these injuries. So this is also something that makes research in nerve regeneration and finding ways to alleviate a lot of those symptoms we just saw on that other list, so that this will lower the cost that the patients incur. Now, to talk about spinal cord injury, I think it will be useful to first go through the spinal cord and talk about why it's so important, what it does, and also why it's so sensitive to injury. So just like we've done for the diseases we've covered before, where we discuss the organ system, why it's so important, what it does, its function, and then we get to the disease, I want to do the same thing for this injury and first highlight the points that make the spinal cord so interesting. So over here we can see the body worlds exhibit image that I really love to share with students, because this shows just how extensive our nervous system truly is. So you guys have seen this in a previous lecture I've given, but again, just as a reminder, this was made by fixing a real human with a special kind of acrylic or plastic. Essentially, they got into their nervous system and they were able to preserve this and see the brain, the spinal cord, and all the peripheral nerves. So we know we have our central nervous system, that's the brain and the spinal cord, and then we have these peripheral nerves that go throughout our entire body, our organs, our skin, our muscles. And we can see that encasing this spinal cord that's exposed in this picture are a whole bunch of bones. These are really, really important for shielding this very sensitive and delicate spinal cord. And we can see that it's broken down by each vertebra segment. All right, so when we talked previously about a cervical 4, a C4 injury, we were specifically speaking to this region of the spinal cord. So when someone has a spinal cord injury, they're able to pinpoint whether it's a T9, L2, etc., and where that was found to diagnose the extent of their symptoms and what they can expect. The cells of the spinal cord are very diverse, but let's first start with the all-star, the one that everybody knows, the neuron. Here we can see our neuron with the cell body, its nucleus. It's surrounded by dendrites that are capable of receiving information from other neurons. We then have the long myelinated axon that ends in axon terminals that make synapses on other neurons, dendrites, or muscles, or organs, or cells that are innervated by neurons. And I use that word innervated specifically because I'll use that throughout this presentation again. Innervation essentially is this process where another nerve innervates something else. It can be another neuron or it can be any other cell. Why we're going to highlight this is because we're going to see that the spinal cord is full of axons that are innervating all sorts of different organs of the body as well as the brain. And we've discussed this before. We know how the neuron works, but just as a quick review, the neuron initiates something called an action potential. Now, this action potential starts from received information at its dendrites that then add together to trigger a chemoelectric signal, which is what we see here. And I have shown you guys some really great YouTube tutorials that demonstrate what an action potential is. But again, we're just going to go over this briefly here, that the action potential is a chemoelectric signal that is carried along the axon. So the axons of neurons are what are transferring this information. The cell bodies are where a lot of our protein production and cell maintenance are occurring. And so again, today we're going to be highlighting how these axons are responsible for the connectivity that we find all throughout the brain. That they are wiring together neurons to send these signals that we see in this computer graphic animation, and that collectively give rise to all of the network activity we see in the brain. And it's this network activity in the brain that will ultimately get transferred out of the brain to our periphery, to our organs, to our muscles, to command the body, to move the body. And it's going to be the spinal cord that's responsible for that. And so, like I said, the cells in the spinal cord are very diverse. And here we can see that the microglial cells, which are resident immune cells, are checking out these other cells. They're much smaller, constantly surveying cells and looking to see if they're healthy, if there's any sort of foreign debris that shouldn't be in the spinal cord. So again, we can think of the microglial cell as an immune cell. Astrocytes are helper cells. Now, I use that term very loosely because for a long time astrocytes have sort of been seen as this sort of just only a helper cell. They're just there to support neurons, but that couldn't be further from the truth. Here we can actually see that these astrocytes are interfacing with ependymal cells of the central nervous system that give rise to the cerebrospinal fluids. We can also see it's interfacing with neurons at all these different points. It even has contact with microglia. So really astrocytes are helping just maintain the homeostasis, meaning just the resting metabolic basal activity in the brain. They're extremely critical for the survival and support of not just neurons, but also their activity. And we can find these glial cells also in the peripheral nervous system. And the peripheral nervous system is obviously intimately tied to the spinal cord, which is the center of our talk today. So it's worth noting these similarities between the central nervous system and the peripheral nervous system that both interface with our spinal cord. And we can see that the cell body of this type of neuron, which is in yellow here, it's a dorsal root ganglion neuron, which also has an axon, is covered in glial cells. These are called satellite cells. And down on these axons, just like the oligodendrocytes that we see in the central nervous system, we have Schwann cells. And these cells are going to be largely responsible for the myelination. All right, so we can again see oligodendrocytes, Schwann cells, and that they are both myelinating the axons of these neurons in the central nervous system and the peripheral nervous system. So I like to think of neurons in the same way that we think about Batman and Robin. Okay, so everyone likes to focus on Batman. Batman's the star of the show. Batman does most of the action. But at the end of the day, we really know that Batman would be in a lot of trouble. He wouldn't have made it out of some really hairy situations, might not have the support he needs, needs that sidekick. And that is how we're going to look at glia. And that we have this young Robin here that's going to help out our Batman or our neuron to do their job, and they really work together. So what are some of these neurons in the spinal cord specifically? Because I say the word neuron, but that's actually a very loose term for a broad type of cells. So here we can see that these chemoelectric capable cells take on many different shapes that are going to help them perform different functions so that they can have different types of synapses, so they can communicate with different types of neurons in both the central as well as muscles and peripheral organs in the peripheral nervous system. So we can see they can be unipolar, bipolar, pseudo-unipolar, or multipolar, and all of these have different shapes. But generally speaking, we can sort of map some of these shapes to function. So on the right-hand side here, this multipolar neuron, we know are going to really consist of motor neurons. So motor neurons have these very long axons, and they are the ones that are actually going to control the skeletal muscles that we find all throughout our body and give rise to movement. Now, interneurons in blue here, they also look like these multipolar neurons, and they're going to be restricted to the spinal cord. So they will not leave the spinal cord the way the motor neuron does to go out to our skeletal muscles. These interneurons will remain within the spinal cord, and they will integrate sensory information to control and coordinate muscle movement and reflexes and sensory feedback. And then to bring up that sensory feedback, we have our sensory neurons. And these are going to be found on the outside of the spinal cord in those ganglia that I just referred to previously in the picture of the peripheral nervous system's glial system. These dorsal root ganglia house a special type of neuron called the dorsal root ganglion neuron, and they give rise to our touch, pain, and proprioception. Now, proprioception, all this means is the body's awareness of where it is in time and space. So if your hand is raised, or if it's behind your back, or whether you're standing up or sitting down, all of this gives feedback and gives us this bodily awareness or proprioception. Now, like I said, glia are very important, and we also have a diversity of glia in the spinal cord. And so, like I said, in order for these glia to perform all these different functions so that neurons can survive, so they can support the neurons and maintain extracellular environment, they are also going to have different shapes to perform those functions. Now, the types of glia that we see in the central nervous system are going to differ from the peripheral nervous system. But in the central nervous system, we have astrocytes, and they're going to maintain the neuronal extracellular homeostasis, and they are also going to activate after there is an injury. They're going to help form something what's called a scar tissue or a glial scar. And then, of course, there's the microglia. I already introduced them as the immune-like cells. They too will activate after an injury. So we'll see astrocytes and microglia contributing something called gliosis after a spinal cord injury within the spinal cord. Then we have the oligodendrocytes that are going to myelinate the central branches of the axons, so the axons that are found within the spinal cord itself, not the axons that are found on the periphery, because that will be done by different type of glial cells. So the satellite cells will help function like astrocytes and maintain the cell body's function, and the Schwann cells will perform a similar function as oligodendrocytes in myelinating axons. So here's the Schwann cells myelinating and the oligodendrocytes myelinating. And I bring up this myelination because, again, for that chemoelectric signal, the action potential to function properly, we need these cells to perform this job of myelination so we can have proper neuronal signaling. To discuss the spinal cord, it's important that we also tie in the different distinctions of the nervous system, because this is going to come into play when we talk about spinal cord damage, just the types of ways our bodies can be affected by a spinal cord injury. So what is it that the spinal cord injury is really doing? What are some general functions it has? Well, very generally speaking, it performs something called autonomic function, or involuntary and unconscious regulation of all the organs and glands in our body. So it's helping everything we have in our body sort of just continue to function and work properly. Now, that can be further divided into a sympathetic or parasympathetic system. And generally speaking, that means sympathetic, fight or flight. We've heard this before. Sympathetic is going to be activated if we're doing things, if we're hunting, if we're moving. Well, I guess these days hunting isn't pretty high up on the list, but maybe playing a sport or trying to rush to class because we're late. That's going to activate things like sympathetic. Whereas parasympathetic is going to be when we're resting and digesting. So it's going to activate a very different system because we're going to need different types of metabolic regulation, different types of activity in these different scenarios. And so, like I said, the spinal cord is going to relay that information. The sympathetic system we can find on the left here is mainly going through the thoracic regions and innervating all of these different organs. And then we can see our parasympathetic system in the cranial and sacral. So for this comparison, we see again that they are innervating sometimes similar, sometimes unique organs and helping to control and modulate their activity in different states. So what are some of those states? Well, we could say, for example, the sympathetic in the eye will dilate, whereas parasympathetic would constrict. Or another one would be the heart rate. Our sympathetic system is going to increase heart rate, whereas parasympathetic is going to decrease heart rate. And some are unique. So unique to the sympathetic system, we have epinephrine secretion, or regulation of the adrenal medulla. The somatic nervous system is our voluntary system, or relating to the somatic, meaning relating to the body. This voluntary system means that we can take control of all our body movements via skeletal muscles. And that's depicted in a very simple diagram here, where the motor neurons leave the spinal cord and innervate muscle groups. And by innervating these muscle groups, they can control when they flex or extend, and all of these things are going to give rise to the movements of a body. And to finally hit this point home, we can see in this diagram that the somatic nervous system, autonomic nervous system are essentially running different processes. Somatic is keeping us moving, allowing us to control ourselves in voluntary motions in our muscle groups, whereas our autonomic system is regulating all the things that keep us alive, like smooth muscle control of the gut, our gland secretions, and even our heart. So we can imagine then that if the spinal cord is relaying all those nerves and all of that coordinated network activity, then an injury is going to affect those processes. So how can the spinal cord become injured, and what exactly does the spinal cord even look like? Now on the left, we can see that the spinal cord has been exposed, so this is sort of like a cross section where we've gotten most of this bone removed in this picture so that we can see what the cord looks like when it resides in the vertebra, and even what it looks like to have these ventral roots coming out, which would give rise to our motor control. Now, on the right here, these very pixelated text that I don't want you to read because you don't need to learn these definitions. This is something you will reserve for when you get to med school or nursing school. But for now, we're really just focusing on the middle here. I just want you to appreciate the anatomy of the spinal cord and that it's even covered in many different protective layers. So here we can see all the different meninges that cover this spinal cord, almost like a protective piece of skin. And then we can also see that nerve roots leave the spinal cord to then go out to our periphery. Now we're looking down on the spinal cord, so if I go back an image, this is going to be an actual cross-section of human spinal cord tissue. And when we go over to the right, we see that shape is right here. All right, so now we're looking down on the spinal cord, and here we can see those dorsal root ganglia. This is where we're going to have sensory information that will come into the spinal cord. Down here is where we will have motor control leave the spinal cord. And all of this bone functions to protect this very sensitive cord. I mean, truly appreciate in this moment all of these layers. We have the bone, we even have fat, and then we have all these meninges that are solely there to protect and encapsulate and also create a barrier, okay, known as the blood-brain barrier, to separate the spinal cord environment, protect the spinal cord environment from the um, external environment. Here is a hand-drawn picture by a Miss Drummond here, who back in 1971, when we didn't have all this nice cartoon and CGI graphics, we had hand-drawn diagrams. And so this diagram is just depicting all of the vasculature that covers the spinal cord. It even goes inside and all throughout the spinal cord. And I just want to hit this point home because this is important to remember. Remember just how much we spent on COPD and why we breathe, just how much time we spent on stroke. So we can even imagine here that this could even lead to strokes in the spinal cord. You can have that happen actually because you have these arteries. So it's important to remember that we have to supply oxygen to all of these very active neurons and their supportive glia. And in order to do that, we're going to have some sort of vasculature in the spinal cord. And so that we can see here. And I'm also including this picture to the right that I've taken from when I was performing a surgery on rats. And here you can actually see an artery that was laying right over top of the spinal cord. That when performing the surgery, I had to be very, very careful that I not damage this because otherwise I would lose this animal because it would bleed out very fast. Now here is the anatomy of the spinal cord. So we saw one earlier, but I wanted to bring up another cross-section of this. This is actual histology, and here we can see in the middle, we're going to have a different shade than we have on the outside, all right? And we can also see roots that are leaving the spinal cord. Now they've been damaged a bit from sectioning this tissue, but you can imagine that if this were to be reconstructed, sort of like deli slices from the deli counter at a grocery store, where you start with the one thick block of meat and then you section it. That's how this was created. And so we can imagine if we put this back together, we would in fact restore a lot of these regions and see that this was one thick solid root. This was the ventral root that again gives rise to motor control, and then here would be the dorsal root that gives rise to all our sensory information and sensory feedback. And that's where I want to start to distinguish some of the finer points of the spinal cord. So we're going to move in here a little bit more. I keep talking about sensory, motor control. Here we can see them working together. All right, so we can see that our dorsal root ganglia have axons that go out to our periphery. They're going to innervate our skin and give us a sensory reception, so feeling, touch, pain. All of those things will feed back into the spinal cord that will then interface with those inner neurons that I were talking about, the inner neurons that were regulating activity in the spine. And they will even go right to motor neurons to go back out and fine tune behavior and movement of muscles based on their feedback from sensory information. So there's this really beautiful networks that exist in the spinal cord to help us be aware of the world around us and really have a lot to do with our sensation and reflex ability. Now, again, I keep talking about distinctions in the spinal cord, and I want to keep zooming in further and further and discussing those because I don't want to just gloss over this stuff. They're really important differences. So we see again, I'm bringing up now the difference in colors. So we see out here there's a lighter pink versus a darker pink. Out here is a lighter gray versus a darker gray. What is that? Why does it look this way? That's because we have white matter and gray matter. White matter will be consisting of all of those axons that we talked about, the long processes that convey and communicate chemoelectric signals, compared to the gray matter, where it looks much darker. It's this dark gray because it consists of nerve cell bodies and interneurons. So all those cell bodies of glia, all the neurons, and then even the interneurons are residing in these regions. And therefore, those cell bodies, because they have special contents in them, we can stain them and they make this cord look different. Even without any staining, you would even be able to see this difference because the cell bodies versus these highly myelinated and thin axons are going to compact differently and give rise to a different look. And one of the ways we can really hit this point home is through crescent violet staining, Nissl-myelin staining, where essentially we're able to stain both the gray matter and this white matter, and they'll stain differently, so that when we zoom in here, we can actually see a nice border. So I chose this region specifically down here where I have this magnifying glass, because we're kind of looking at that region right here. You can imagine that border would go right through where this laser pointer is going. So we can see down here what white matter would look like, where it's mainly just these axons and some glial cells. And then up here where we have gray matter, where there are these large neuronal cell bodies that stain very densely for this Nissl. This is something that's going to stain different organelles that are found uniquely within the cell body. And that's how we get the distinction between the gray matter and the white matter, because this is where all the cells are, and this is where all the information carrying axons are. And this is relevant for a spinal cord injury, because we can imagine that where we injure, if we were to just injure slightly here with maybe a contusion, versus an injury that would make its way all the way into the deep gray matter and affect these cells. So I want you to think ahead to injury and how these different regions may react differently to injury. Now, even within the white matter and gray matter, we can further subdivide. Now, in the white matter, we can see that we have different types of axonal tracts, okay, these tracts. So I want you to think of these long axons that would be ascending. So axons would actually be rising up and out of this slice towards the brain or towards other regions of the spinal cord. And we can see that there's many different areas like that. The dorsospinal cerebellar tract, this is the fasciculus gracilis. All of these different types of tracts are going to be giving rise to our sensory afferents, or they're going to go toward the brain. That's what afferent means, towards something. And in this case, the reference point will be the brain, our central command. And so these sensory axons are going toward them. That is in comparison to the right side. Here we can see the motor pathway or efferent pathways are actually even anatomically localized in different spaces. They're sort of opposed, right? The ascending dominates the top portion or the dorsal portion of the spinal cord, whereas on the ventral side we have a lot of our motor or efferent pathways. And so these guys are actually going to be coming from the brain, or efferent meaning traveling away from the brain and going out into our periphery, out from the ventral horn to control our skeletal muscle. So again, thinking ahead to injury, we can imagine that an injury on the dorsal side versus the ventral side would give rise to different injuries, right? One mainly affecting maybe sensory information, the other affecting your ability to control your motor function. And this is depicted here again, just so you guys get the idea that we're looking at one slice, and I kept rising my laser pointer up like this. So here we can see many different sections that go from sacral to lumbar, thoracic, cervical, all the way up to brainstem, so that we can appreciate that some of these axons are going all the way up. Traveling all the way to the brain to allow us to understand different pain, touch, proprioception, and give feedback to our brain about what our body is sensing. Compared to the descending pathway, where we see again most of this residing in the ventral region, but again similar to the ascending pathway, for the descending pathway of these long axons that are coming from the brain, traveling all the way down our spinal cord, and at different regions they will exit to give function and motor control to different parts of our body. And so we can think about this again when we think about paraplegia, tetraplegia, and where in the spinal cord an injury occurs, and why that would then change whether or not you're able to control your arms or your feet or both. And very briefly, when we talk about these connections, or when I speak about the peripheral system and innervating skeletal muscle, we can see here that we are getting into all these different muscle bundles from the ventral, and this is from our motor neuron. And these types of synapses are going to be called neuromuscular junctions. But it's important to remember too that we have synapses within the spinal cord, all right, and within the brain. So these synapses are going in both directions. They're going out and they're going deep, deep, deep within the brain. Well, I guess it's important to note too, and in some instances, from deep to also out into our cortex. All right, and just briefly, I mentioned the word synapse, so I quickly want to review it. We recall that a synapse is just the junction between two different cells. All right, and specifically, it's going to be a neuron to another neuron where we have a presynaptic and a postsynaptic cell, and the presynaptic cell is going to release neurotransmitters that can then bind to receptors on the postsynaptic cell. And our spinal cord is full of these. Now, before we dig into the finer points of a spinal cord injury, I just want to quickly review what touch and motor control actually looks like. Let's imagine that we're trying to set the temperature on a shower. You can see that we have a sensory ending in our skin. We'll have many of these, but in this case, we're just showing one that would respond to the temperature of the water. This temperature feedback would travel along this axon of a sensory neuron. It would be traveling via its action potential, which again is that chemoelectric signal. Here we see we're passing that dorsal root ganglion neuron, which is the axon of this neuron, and it is then going into and entering the dorsal horn. Once it enters this dorsal horn of the spinal cord, it then enters our ascending pathway. Notice that we're in the white matter, so this is going to ascend up the spinal cord, where its next stop will be to enter the spinal cord and synapse with the brain. All right, so we have our first synaptic connection with the brain here. It's going to relay the original temperature of this water into the brain pathway, and this sensory pathway will continue with a second neuron in the thalamus, which is a region even deeper inside of the brain. And this sensory pathway will reach the cerebral cortex for conscious perception. So from the thalamus, we will have an axon that goes out to our cortex where we have a lot of our conscious awareness and a lot of our conscious calculations, if you will, that will then send some sort of control or response. So was that temperature good? Was this cold, too hot? What do we need to do? Do we need to turn a knob? Are we just good? So then we'll see here this upper motor neuron from the cortex is going to execute a motor command. And so this is actually going to send an axon all the way through the brain, down our spinal cord, and interface with motor neurons within the ventral horn of the spinal cord. It will then leave through the ventral root and make its way to the muscle groups of the hand to either retract away and start to adjust these knobs or withdraw and go ahead and enjoy that shower. So we can hopefully appreciate that we've integrated all the previous slides I just walked through and shown just how it is that our body not only senses the world but also interacts with it, and that it's a very intimate connection between these two systems. In fact, it's so intimate that we have many different reflex pathways that don't even require the brain. So again, these pathways will not require brain control. So how this is going to work is we're going to have our skin, our dorsal root ganglion neuron axon is going to receive information from the skin. It will travel along its axon into the spinal cord, interface with motor neurons or interneurons that interface with motor neurons, and then go down and control muscle cells. So for example, on the right, we can imagine if someone were to touch a hot flame, this temperature of the flame is going to travel through the sensory axon, the dorsal root ganglia axon, all the way in to our dorsal horn. It'll interface with some interneurons, strong signals, because this is a really strong and intense reaction here of heat, that will then go into these motor neurons, travel all the way down to quickly trigger a reflex. So pain receptors are intimately tied with a reflex pathway in our spinal cord. So that's the pain response. It's why we don't leave our hand on a hot stove. But here's one that's a bit more medical that we're probably familiar with from a doctor's office to test our reflexes, where here we can see a tap to the knee creates a reflex in the leg muscles using a very similar reflex pathway. So why I wanted to bring that up is because the spinal cord we can appreciate now is not just being a relayer of information to the brain or a relayer of information from the brain to the periphery, but we also in that last slide just appreciated that the spinal cord itself appears to also have pattern generation, reflex generation, essentially what we could think of as spinal circuits, unique and independent spinal circuits. So that makes the spinal cord extremely important. But unfortunately, there is no therapeutic treatment for human nerve injury. And I am very specific here to state human nerve injury because that's not just limited to the spinal cord. As we're going to learn next week when we talk about traumatic brain injury, we'll see that there is really not much that we can do for injured nerves. And why is that the case? So let's just real quickly talk about what is a nerve injury. Now, we could base a whole class around this. I could spend a whole bunch of time, maybe even a semester on this. This is my field of research, so it is something I'm really close to. But I want to communicate this and keep this in the realm of this class. So I'm really just going to put this very simply here in this diagram that we can think of a nerve injury in these simple steps. All right, so we have a knife in this example. So we're going to cut the axon. There could be many ways we could injure an axon. It could be with a crush, a pulling, tearing motion. But in this case, we're just going to have a nice smooth cut. Within minutes of this cut, there is a molecular cascade of events that occur, where we have things responding to influxes of molecules such as calcium to help sequester them and start to heal the region of the axon that has just been injured. Most importantly, because we can realize here that this axon will connect back to the cell body, whereas the distal axon is connected to nothing. Over time, this distal axon will actually begin to degenerate. This is a process known as Wallerian degeneration. And over to the left here, we're actually going to see an attempt at some regeneration. So we're going to see a sealing off, survival, a persistence. There will be some dying back, okay, initially of this axon. But if we give it some time, sometimes, even in most cases, you'll see a little bit of attempted regeneration, some sprouting or some end activity here on a neuron. And specifically what we're talking here is in an adult system. So post birth, fully developed human, or we now have this axon incapable of mounting a really strong regenerative response. Over here, we can see the formation of a glial scar. So like I said, the central nervous system is full of a whole diversity of different players, different cellular types. And here we can see those astrocytes that we mentioned that help cells, they become reactive. And meanwhile, both the microglia as well as an influx of invading macrophages from the periphery, they're going to be hard at work to clear this debris of dying and damaged tissue. So really, in a crash course here, you got the basics of what a nerve injury is. We have this molecular events that occur within the cells to try to react to the injury. We have a very, very feeble and weak response to that injury, some feeble attempts at regeneration. But then we have all of these other cells that hop into action to help quarantine the site of injury. And this, overall, the glial scar and this weak capability of regeneration is what makes spinal cord injury so difficult to deal with, and why there's no treatment or cure, and why many of these people will not regain function in certain parts of their body. So what is it that's preventing an adult axon from regenerating, and has anything worked? You know, I just made that sound so hopeless. I am actually very excited to work in the field of regeneration because I remember when I first got into neuroscience, which was actually 2006. It was years ago, where it was still being debated whether or not adult neurons even regenerated. And now we know that we can get adult neurons to regenerate. In fact, there's many different models that have done this. And so one of the barriers that neurons face is something called an intrinsic inhibition. Intrinsic meaning within the cell itself, that there's a block. There's some incapability of regenerating, and that's because adult neurons, they're no longer growing. They're no longer going through development. They're no longer receiving signals that make them extend axons. So researchers have thought, hey, if we could tap into those pathways, if we could make an adult neuron think it's in the need of growing and find a way to tap into growth promoting pathways, can we get them to regenerate? And sure enough, researchers have done this. We can see here some amazing work by Dr. Kevin Park even back in 2008, where they showed that through knocking down a protein called P10 that regulates metabolism, that they could get these axons to regenerate robustly within an animal model of an optic nerve injury. Now, there are also extrinsic barriers to regeneration, and those would be the glial scar. So like I mentioned previously, the astrocytes and microglia after an injury are going to become reactive, and they will give rise to what's called a glial scar, which is working to quarantine the site of injury. And researchers have actually isolated enzymes from bacteria. All right, chondroitinase actually comes from a bacteria, and they were able to apply this and degrade some of these chondroitinases. So this chondroitin sulfate proteoglycans, or CSPGs, that make up this glial scar, they can actually use this enzyme to degrade them. Now we can see in this top picture that some of this was successfully degraded, whereas other variants were not. So there's still some way to go with this. There's an amazing work by Dr. Jerry Silver who works on this, who's shown some really, really promising results that clearing up some of this glial scar, or at the very least making the glial scar environment more permissible to regenerating axons, we can restore some functional recovery. All right, so I want to kind of switch gears and talk about why adult regeneration is so difficult. And that's because during development, we have some amazing, amazing events that are going on. And so I'm introducing you guys to this man to the left here, Roger Sperry, who in the 1940s did some really cool experiments looking at this. And so this is a review from 2007, where we're looking at just exactly what it was that Sperry was messing around with in the lab. And so this can seem kind of bizarre, but honestly, this kind of work has really given rise to some amazing insights in developmental neurobiology and how our nervous system is wired together from the start. So I want you to imagine a frog and a frog's retina. And what researchers found, if they were to inject dyes or tracers into the retina, they could find that retina that were found in region 1 or region 2 or region 6 corresponded to a region deeper in the brain. So axons would leave this region of the retina and go all the way into a region of the brain called the tectum that was topographically equivalent to the type of pattern that we saw in the retina. So 1 would always go to 1, 2 to 2, 8 to 8, etc. So this matching or this directed development really was a head scratcher for Sperry, and he wondered, what would happen if we just took this developing eye and rotated it. So if they were to cut the optic nerve, rotate the eye, suture it back together, and then let development finish its course, would 1 still go to 1? So now if we've twisted it, 1's on the bottom, whereas it used to be towards the top. And so now if 1 is on the bottom, 3 is on the bottom, will 1 still go to 1, or will 1 end up at 7, for example? So what they found when they did this, and so here to the left we can see these tracers, is that when they rotated this eye and they presented this frog with a fly, they found that with the rotated eye, even when the fly was on this side, the fly was shooting its tongue in the opposite direction. Now, why that is such a eureka moment and was so profound to Sperry was because he realized when he opened up these frogs, and also it complements the behavioral response, that 1 was still going to 1, and 2 still to 2, 5 to 5, etc. So the position of a cell in the retina was enough to guide the cell to a specific point. And that's what gives rise to this graphic that we see here. So a collection of cues in the developing frog eye, chemo-repulsive cues and chemo-attractive cues, as well as surface contact repulsion and contact attraction, these long-range and short-range cues collectively were helping guide the axons of the cell in position 1 to a position 1 in the tectum. Now, all of these cues collectively give rise to gradients, and that's what we want to appreciate by these different gradients depicted here of different secreted proteins, and down here different expressed types of receptors. And we are appreciating down here that different retinal cells are going to project their axons and be attracted to very specific positions within the superior colliculus or a deep brain structure of the rodent brain based on what molecules are being secreted by target cells and the types of receptors that these retinal cells have to be able to detect those signals. So cells that really favor a strong expression of the Wnt and ephrin, but a low expression of ephrin, are going to want to target over here towards the left side, whereas the ones that favor a high expression of ephrin and a low expression of Wnt are going to want to favor the right side. And it's these gradients that allow our nervous system to finely tune where axons go and how the entire network is connected. And this is really just such an amazing and beautiful thing to appreciate about developmental neuroscience. Now, how is it that these neurons are able to extend these axons to such specific points? I was talking about these secreted molecules, and then I was talking about receptors that the neurons' axons needed to obtain or have. So what is that? What am I talking about? And where would those receptors be located? Those receptors would be found on something called a growth cone. So here what we can see is a growth cone coming out of the axon or growing axon of a neuron, and we see it has a very unique type of structure. It has these little filopodia, a larger lamellipodia, and really this thing is working as an almost sensory motor function to survey its environment and extend new axon. And in fact, we can see that happening in real time in these images to the right, where we can see now structures called microtubules. So these filamentous, very dynamic, long little fibers that are glowing here, these are called microtubules, and they are going to be responsible for pushing and growing and ultimately laying down new axon. So here we can see axon, and this axon is going to be elongated as this structure moves forward. And these pictures are compliments of Hemalata, who was a previous lab member of my lab. Dr. Boss's lab, where these images were taken, and we studied microtubules and how they could enhance regeneration after an injury. Now here we can see the growth cone is going to function to move into a favorable substrate. So first it's going to encounter this substrate, so there'll be receptors that are capable of receiving this signal and realizing that the substance is there, and that will send a signal internally that will mobilize this cytoskeletal structure. So in red we have actin, in green we have our microtubules, and we can see that actin and microtubules will work together to invade this favorable substrate. So as these signals that are being triggered on the surface of this cell from this substrate mobilize the cytoskeleton, you'll notice that the growth is mainly directed towards the favorable substrate and not where there is a lack of the substrate. And over time, we'll see engorgement. So many of these microtubules will begin to polymerize and fill this growth cone, so that then we have a new axon shaft formation as this consolidates and the whole process continues, and this growth cone will continue to move towards a favorable substrate. So when we were talking about those retinal axons in the frog and in the mouse eye, we're appreciating that they, during development, would have this structure to guide them to their target cells so they would function properly. And this is what I meant by the receptors that you would find on them, that these receptors, every different neuron is going to have some different combination of these receptors. So there's going to be some non-homogeneously distributed cue in the environment. That's that gradient of signals. Then there's going to be cell surface receptors that can bind and respond to that cue. And they're going to ultimately decide where and how the growth cone maneuvers, because it's going to integrate signals with the cytoskeleton that help this thing turn and shift and move about an environment to make its way to a target. Now it's worth noting that all of this axonal growth is to have this event occur. We want a neuronal network. We want our developing neurons to have their axons properly find their targets. Okay, now whether that be another neuron or in the case of a peripheral cell, a muscle tissue or organ, we want this connection to occur because the axon will then be releasing neurotransmitters at the synapse to affect activity. And that's going to give rise to all of these different connections. I'm showing you guys this so you can just appreciate that we're often just showing these very direct, kinds of one-to-one connections here, but really connections can be absolutely crazy. So we've talked about topography today, where retinal cell 1 matches to retinal cell 1b in some other region, and there's this sort of direct connection. But this can get really, really complicated. We have divergence, convergence, lateral inhibition, but all of these different types of connections are going to give rise to the really complex connectivity that we have in the brain that can give rise to all these different forms of patterned activity. And so we're really right now appreciating that during development, all of this is occurring and connecting. All right. And so hopefully you're already thinking ahead to injury, that after we have an injury, we can imagine how do we tap back into this growth promoting pathway. So a metaphor I have to kind of hit this point home is this just slew of wires that you would find in like a place where you have network activity, maybe even in a college campus, your IT department, so everyone can have networks and Wi-Fi. They might have a room like this. And so I want you to think about the brain in a similar way, right? I want you to think that somebody one day came along and set all this up. Somebody who knew what they were doing, okay? And we can compare that to the developmental process that happens when an organism's growing. It was directed, it was specific, there was order, there was a sort of guide to the whole thing. But then, if you imagine an injury comes along, or in this example, if I just start unplugging some wires and then ask you guys, hey, can you do me a quick favor and just plug those back in? Plug these wires back in. How do you know where they went? How do you guarantee they get to the right point? And so that's why I took the time to show you guys and talk about guided axon growth, because that's the problem with the barrier to adult regeneration, is that one, the neuron's no longer in a state of growth control, so they don't have a large amount of growth pathways active and growth promoting pathways going on. And then two, all those signaling molecules that exist that guide the axon to the appropriate target, they're absent. They're not there. That whole system is no longer online. So you're trying to get this axon to regenerate in an environment, and it doesn't know where to go. All right, and then in this image, we're going to see our first introduction to the glial scar proper. So this is a microlesion. This would be a very fine injury to the spinal cord. We can see that there's this altered ECM. So we have our key over to the side here, this pink area. This altered ECM will be molecules that are secreted by different types of macrophages, and to some extent some of these astrocytes that are in the line of this injury. Now appreciate that when this lesion occurred, it disrupted these axons. So this dystrophic axon now, this is not a cell body, this is the end of an axon. So the cell body would be way down off the picture somewhere. We can see that these axons, they want to extend beyond this, but because they have this lesion now, this material of macrophages and altered ECM are actually going to work as a barrier to regeneration. So it's going to stop these axons from passing through. Now, in an even more severe injury, we start to appreciate just why the glial scar is doing this. Now, yes, we see again that this is going to be a barrier to regeneration, but in this case of a contusive lesion, imagine that, this was from some trauma incurred to the spine, where it presses in, and this contusive injury, sort of like a bruise or a deep push or press against the spinal cord, is now going to give rise to a really big glial scar. And we can see all these different reactive astrocytes are quarantining this site of injury. So really what the reactive astrocytes are trying to do is stop the spread or influx of foreign tissue or necrosis or the damage that's going on here, whether it's bleeding or entrance of these external macrophages. The astrocytes, they don't want this to get into the rest of the spinal cord. So in some ways, the glial scar and these reactive astrocytes are really working to quarantine the site of injury. And finally, we can see an even worse injury where now we have a stab. We can see that these reactive astrocytes are even more numerous. Now we have the invasion of some fibroblasts as well as the macrophages. So again, depicting, notice the way that these astrocytes are oriented. Yes, they are blocking regeneration, but again, this isn't happening to block regeneration. This is happening to seal off and preserve the tissue of the spinal cord that is left behind. So while yes, this is a barrier to regeneration, what the take-home point from these last three images were is that the glial scar is performing a function. The glial scar is actually protecting the site of injury. If we were to get rid of these reactive astrocytes, which researchers have tried to target, if we were to try to stop the glial scar altogether, then we'll actually find that we make injury far worse. So we do need some glial scarring, but the question is, can we break some of this up a little bit to allow some of these axons to pass through? All right, so this brings us towards the end of the lecture, and it's going to introduce the topics that we'll be talking about during our class discussion. So I just want to introduce you guys to some very exciting experimental therapies that are ongoing. So the first one here we see is epidural stimulation. Now, for an epidural stimulation, what we want to appreciate is this is a surgical implant that will be installed into a person's body with electrodes that lay across. Now, there are some others that actually do enter the spinal cord, but for the most part, epidural stimulation means that we're going to have that lay across the dura, and it will then be able to stimulate spinal nerves based on this pulse generator. Now, the take-home idea of all of this, in short, is that the pacemaker will be able to, or I'm sorry, the pulse generator will be able to regulate activity and feedback activity to control things like heart rate as well as bladder control. There are some really terrible things that can happen in connection to bladder and heart regulation, where every time your bladder fills, your blood pressure can rise, and it can actually be a very lethal side effect of spinal cord injury. So epidural stimulation is being looked at as a way to help treat that. Now, what you're seeing in this GIF at the bottom is a individual who suffered a spinal cord injury, and you can see had lost motor and sensory function of their legs, and yet here in this apparatus are capable of stepping and walking. Now, yes, this is keeping this individual upright and holding some of their weight. However, what we're really appreciating here is that this individual is stepping. This individual is walking to some degree, and this is all via this epidural stimulation. And we're going to talk about how the spinal cord consists of these spinal networks that maybe one day, far down the future, this technology could really improve, and we could potentially, without the need of a brain, directly regulate the tissue that's spared below a site of injury and restore some function. Stem cell therapies are another promising experimental treatment for spinal cord injury. The idea is that the stem cells will be used to create other types of cells, neurons, oligodendrocytes, astrocytes, that will then be used to either bridge the gap or act as a relay for the injured tissue. Stem cell technology has come a long way. There used to be a lot of controversy over the source of stem cells, which was from fetal tissue. There was also found that there were stem cell niches in adults and that they could be obtained experimentally. Now, today, stem cells are obtained from many different means. One of those is to derive adult stem cells from the somatic cells found on adult tissue. So even just taking skin cells, they can be induced or reprogrammed using viruses and known genes that are important for stem cell states to then create a stem cell population. And these stem cells are further differentiated into the target cells, which are neurons, oligodendrocytes, and astrocytes that can be transplanted into an injured patient's tissue or cord. Now, why would we do this? Why are these stem cells going to be transplanted, or better yet, why are the differentiated stem cells, so those neurons and glial cells, why will they be transplanted into an injured tissue? Well, when we talk about bridging this gap, here is a depiction from a nice review written by Asink et al. 2017, where we can see bridge formation. And these purple cells here are going to act as the transplanted bridge that then in blue, these injured axons are able to grow across. So in a previously unfavorable environment that contains the glial scar and repulsive cues, we can now put in cells that are favorable and can be conducive to the regeneration of neurons, specifically their axons. And so we can think of this like a bridge. Here's a famous bridge in Philadelphia, and so we're going to bridge the gap of tissue and allow for these axons to cross. Another way we can think of stem cell therapies are their potential for acting as a relay. So the transplanted cells that have been induced to become a neuron, meaning that these stem cells were differentiated into a neuron, they can be transplanted into the tissue, and now the injured axons will make connections with these transplanted neurons. And the idea being that, kind of like an extension cord, the transplanted neurons will then be capable and regenerative capable to extend an axon and grow and potentially make connections upstream from the injured tissue, so that now tissue that was no longer connected to an upstream environment that still had the connections with the brain, now they may make connections. And so through physical therapy, there is the potential to restore unique circuits in the spinal cord. All right, so for your guys' assignments, you're going to review a Nature article that discusses epidural stimulation, and then you will read a clinical trial, the CELLTOP clinical trial, which is a first report from a phase 1 trial of stem cell therapy for traumatic spinal cord injury. This was a N of 1, so really one patient was used for this, but it'll be an interesting trial to discuss in class about clinical trials, as well as the promise and also controversy of stem cell therapies. All right, thank you for listening.